Good afternoon and a warm welcome to this presentation, which focuses on the importance of the reference material choice for your PET analysis. When we were performing phosphatidylethanol analysis in the laboratory, we observed that for calibration and quality control samples, when using reference material from Avanti Polar, that the concentrations we obtained were matching quite well for both monitored transitions. However, this was not the case when we analyzed authentic samples which contained naturally produced phosphatidylethanol. There we saw that one of the transitions scored a significantly higher concentration than the other transition. At first, we could not explain what is causing this issue and how we could solve it. However, followed by a conference visit and some internet research, the publication by Katerina Wozny and her group came to our attention. They described major differences in relative abundances of the two fatty acyl anion fragments of phosphatidylcholine, which they observed in tandem mass spectrometry experiments. They state that there is a preferred but not unique collision-induced fragmentation site at the S and 2 positions of the acyl glycerophospholipids. Based on that information, we guessed that it could be that the observed effects for phosphatidylethanol are caused by impure reference material. We assumed that it is possible that the ordered PEth-16018-1 from Avanti Polar lipids is not just PEth-16018-1, but also a certain amount of PEth-181-16O. Within the fragmentation cell of a tandem mass spectrometer, this could cause differences in fragmentation pattern as indicated on this slide, which could then lead to quantification differences. To study if that assumption is true, we obtained PEth-16018-1 reference material from 3 commercial suppliers, from Avanti Polar Lipids, from Chiron, and from Cerilliant. This allowed us the study of regioisomeric purity by testing CID coupled with ozone-induced dissociation. This was performed by the group of Stephen Blanksby at the University of Brisbane in Australia. In our laboratory, we investigated the fragmentation in the tandem mass spectrometer itself, studying product ion ratios and the impact of the reference material choice on the quantitative result. The regioisomeric purity testing using CID coupled with ozone-induced dissociation and performed by Stephen Blanksby revealed that only 1 out of the 3 reference materials show high purity. For Avanti Polar, we only observed a purity of 75.8%, for Chiron a somewhat higher purity of 79.3%, and from Cerilliant a purity of about 99.2%. Measuring the product ion ratios with different collision energies from 0 to 60 volts in our Shimatsu 8050 mass spectrometer showed that we can distinguish regioisomerically pure reference material from impure reference material based on this product ion ratio. It is indicated here with the dotted red line between 0 to 60 volts for all of the 3 reference materials. We can see that at about 30 volts for Cerilliant, which is pure, we have a ratio of about 30. For Chiron, we have a ratio of about 45, and for Avanti, a ratio of about 50. when comparing the product ion ratios from reference material with authentic samples, we observed that the two impure reference materials, the one from Chiron and the one from Avanti, have much higher ratios than 10 randomly selected authentic samples. However, when comparing these 10 authentic samples to the pure material or almost pure material from Cerulean, we saw no real difference. We therefore concluded that the authentic samples have a very high purity of phosphatidylethanol. They mostly contain 16O-18-1, and that purity is a key parameter in order to match these samples with commercial reference material. When we then went on to the quantification of the authentic samples and compared what we obtained as result as concentration, we saw that there is a minor to no difference when comparing the MRM1 transition from Cerulean with the MRM2 transition. However, when we compared the MRM1 and MRM2 transition from Avanti, we saw that there is a huge gap. The same was observed for Chiron, however the gap was somewhat smaller there. This means that in order to match the right concentration during phosphatidylethanol analysis, you have to take the right reference material which has a purity similar or identical to the authentic samples which you are analyzing. In summary, when using regioisomerically impure reference material, you observe a mean difference between transition 1 and transition 2, here described as MRM1 and MRM2, of 30% in the case of Avanti Polar and 26% in the case of Chiron. Overall, when choosing your transition and when using impure reference material, it can have a severe influence on the analytical results, and therefore we should be very careful when applying threshold concentrations with non-regioisomerically pure reference material. As a take-home message and to summarize the presentation, I would like to show you the following slide. We see the fragmentation pattern of pure PEth-16O-18-1 and the fragmentation pattern of pure PEth-18-1-16O. Within the red frame, we see the fragmentation pattern of the non-regioisomerically pure Avanti Polar reference material, which is sold as PEth-16O-18-1, but which is actually a mixture of 76% of PEth-16O-18-1 and 24% of PEth-18-1-16O. This means that for the 255 fragment, we have a signal intensity increase, whereas for the 281 fragment, we have a signal intensity decrease. If you use the signal 255 for your calibration and then reference your authentic samples based on these signals, you get a concentration within your samples which is underestimated. On the other hand, if you use 281, you get an overestimation of the concentration within your authentic samples due to the fact that the calibration does not match reality. With this, I'm at the end of my presentation. I thank you very much for your attention, and I'm open to answer your question regarding the regioisomeric purity of reference material. Thank you very much.